Assalamualaikum. My name is Kaina Shivas from MS Chemistry, Batch 7. I will present a review article on crystal silicon based sensors and its application in medical device manufacturing, submitted to Dr. Hammad. First of all, there is abstract. The main points written in this abstract are: because of the silicon unique properties makes it excellent material for creating various sensors, including crystal silicon strain sensors, which rely on their piezoresistive property. These sensors are the key components in advanced medical devices and are the main focus of this review. One critical application is in their preclinical testing, particularly for detecting drug-induced cardiotoxicity. This study basically introduces an SU8 cantilever with a single crystal silicon strain sensor designed in such a way that it can measure tiny changes in cardiomyocyte contraction forces during toxicity tests. This device aims to improve the accuracy and efficiency of drug screening by capturing real-time, small-scale changes in heart cells. The basic features of this device can be their high sensitivity. Because it detects extremely small forces like 0.02 micronewton with a sensitivity 17 times greater than traditional metal strain sensors. The second feature of this device can be real-time monitoring because it enables continuous measurement of cardiomyocyte contraction forces, making drug testing more precise and efficient. In this article, we will not only make this device, we will also experiment it by culturing cardiomyocytes in vitro with their contraction forces continuously monitored. To validate this practical application, cardiomyocytes were treated with two drugs, verapamil and isopropanol. This is all about abstract. Now we will move towards the introduction. The basic points or main points written in this introduction. Basically, first of all, the first paragraph mentions that why silicon is preferred over other materials. First of all, the reason can be first reason can be their exceptional atomic structure. Silicon atoms are arranged in a diamond-like cubic lattice, which provides excellent charge carrier mobility and piezoresistive properties. This makes silicon highly sensitive and reliable for sensing applications. The second reason can be customizable electrical properties. Silicon's conductivity can be controlled by doping, adding small amounts of elements like boron or phosphorus to achieve the desired performance. The third reason can be when silicon crystal lattice is deformed by an external force, its electrical resistance changes. This phenomenon basically known as piezoresistive effect, and it is a main basis for silicon strain sensors. Even the slightest deformation in the crystal structure leads to measurable resistance changes, enabling high precise measurements. Now we can say that silicon is very important in sensor technologies, and with the help of silicon many sensors are prepared, but in this article we will use particularly strain sensor, crystal silicon strain sensor. And the advantages of this crystal silicon strain sensors can be their superior sensitivity. High piezoresistive coefficient ensures that small deformations result in significant resistance changes, and stability and reliability of this sensor. The uniform lattice structure offers consistent performance even under harsh conditions. And their high mechanical strength, single crystal silicon withstand varying loads and provides durability. Their low thermal distortion minimized because this sensor minimizes errors caused by temperature changes, making it ideal for precise applications. Now we'll see this silicon strain sensor application in medical device. Improving the measurement of cardiomyocyte contraction force in drug testing by integrating a highly sensitive single crystal silicon strain sensor into SU8 cantilevers. This is the application. As we know the main points of introduction now. In this application, first of all, we see that why this device is particular for cardiomyocyte. Cardio means heart, for heart. First of all, in this first paragraph, there is mentioned that cardiovascular diseases are the major global health concerns identified by the World Health Organization as the leading cause of death in 2019. Drug therapy is the most common treatment for cardiovascular diseases, but developing new drugs is challenging due to the long trial times and large trial sizes. Therefore, faster and more accurate preclinical drug testing methods are urgently needed to predict drug-induced cardiotoxicity. But the current challenges comes in this but the current challenges in measuring cardiotoxicity can be the first one is batch clamp method, because it is standard for assessing cardiotoxicity at a single cell level and it is limited by technical complexity, invasiveness and inability to handle large sample sizes. The second reason or challenge in measuring cardiotoxicity can be microelectrode arrays, because it measures electrical activity but don't capture the mechanical contractions of cardiomyocytes. So it is to overcome these challenges, there should be advancement in the cantilever based systems. So cantilevers with strain sensors are promising solution for measuring the mechanical contraction forces of cardiomyocytes. Materials like PDMS means polydimethyl siloxane and PI polyimide and SU are commonly used for cantilever because they support cell culture and provide measurable strain. Metal strain sensors, though initially used, have low sensitivity and limited resolution. That's why silicon strain sensors are preferable. Silicon strain sensors offer exceptional sensitivity, stability, and stretchability. These sensors are basically integrated into SU8 cantilevers to measure contraction forces with high precision. This is all the points written in this first 1-2 paragraphs of application. Now we will move towards the structure and fabrication. Basically, I will not read all the review article because it has so much detail. I will tell the main and basic points in every heading, first such as in structure and fabrication. First of all, look at this picture carefully. In this figure basically demonstrates the integration of single crystal silicon strain sensor with an SU8 cantilever for measuring cardiomyocyte contraction forces. Figure 1a basically in figure 1a basically there is a concept of relaxation and contraction. Relaxation is basically when cardiomyocytes are at rest, minimal forces applied to the sensor, and contraction is when cardiomyocytes contract, they deform the SU8 cantilever, which changes the silicon strain sensor's resistance due to the piezoresistive properties. This resistance change is detected as an electrical signal. In figure 1b and 1c, there is an electrical setup. The silicon strain sensor is integrated into the cantilever and connected with the help of Wheatstone bridge circuit. This circuit basically converted the electrical signals from the cantilever deformations due to the cardiomyocyte contractions into measurable voltage outputs. This helps in quantifying the contraction forces. In figure 1d, there is a layered structure of cantilever. The cantilever is composed of multiple layers for optimal performance. First of all, there is SU8 layer, which ensures flexibility and biocompatibility with cardiomyocyte culture, and the chromium and aurum means gold layer provides electrical connectivity, and silicon sensor layer detects deformations caused by the contractions. There's dimension of cantilever can be 6 mm multiplied 2 mm, designed to maximize the displacement for force measurements. In figure 1e, there are microgroove patterns. Microgrooves with width 3 micrometer, depth 2 micrometers are added to the cantilever surface. Purpose of these microgrooves: these grooves basically encourage cardiomyocyte alignment and improve contraction force measurements. Now we Now we see the diagram explanation. Now we will move towards the fabrication process of the silicon strain sensors. Fabrication process of silicon strain sensor. First of all, in first step, a pre-doped silicon on insulator SOI means wafer was prepared with the 300 nanometer thick silicon dioxide layer using thermal oxidation. In second step, the SiO2 layer was etched means produced using buffered oxide etchant and photolithography to create a desired sensor pattern. Basically, photolithography is used to get the desired patterns. Number in third step, tetramethyl ammonium hydroxide was used to wet etch the silicon strain sensor, followed by the hydrofluoric acid 49% treatment to remove the buried oxide layer. In fourth step, a photoresist was applied beneath the sensor for support. The sensor was then submerged in HF to dissolve the buried oxide layer, releasing the silicon strain sensor. Now, in the final step, the sensor was cleaned, dried, and transferred onto a PDMS stamp. As we know, PDMS is polydimethyl siloxane stamp, soft baked at 95 degrees Celsius and pressed into a 2 micrometer SU8 adhesive layer. The PDMS was then carefully removed to transfer the sensor onto the adhesive layer. This is the steps for the fabrication process of silicon strain sensor. Now we will see the how silicon strain sensors are integrated with SU8 cantilever. First of all, a polyvinyl alcohol sacrificial layer was been coated on a glass wafer. Then a 0.6 micrometer thick SU8 film was applied to prevent polyvinyl alcohol dissolution. Then a 12 micrometer thick SU8 layer was patterned using photolithography. An additional 2 micrometer thick SU8 layer was coated onto the cantilever to attach the and strain sensors, silicon strain sensors. Then the silicon strain sensor was affixed to the cantilever with the help of transfer printing technique. Then a 10 nanometer chromium layer and a 200 nanometer gold layer were deposited using electron beam evaporation, where etching and positive photoresist were used to define the Wheatstone bridge circuit. And microgroove patterns. Microgroove patterns are drawn. A 2 nanometer thick SU8 layer was patterned with microgrooves 3 micrometer width, 2 micrometer depth for cardiomyocyte alignment and functionality. And in the last step, there is cantilever release means the cured SU8 cantilever. Then in the last step, the cured SU8 cantilever then on the polyvinyl alcohol layer was carefully cut and immersed in deionized water to dissolve the polyvinyl alcohol sacrificial layer, releasing the cantilever. Then this cantilever we prepared is integrated with silicon strain sensor. Now, in the starting of application, we see the details of the diagram and fabrication process of sensor and this integration with cantilever. Now a device is prepared, and now in the second step we will culture cell culturing step in which cardiomyocytes are cultured. The details or main points can be now the cell culturing of cells. Basically, culturing of cell to cell culture is a process of growing cells from plant or animal in a controlled laboratory environment. The basic points mentioned in this are cardiomyocytes were taken from 1-day-old neonatal rats using a standard method to help the cardiomyocytes stick better. Each cantilever was coated with the fibronectin for 1 hour. Then the cardiomyocytes were placed at a density of 1000 cells per millimeter square on the SU8 cantilever and kept in a regular incubator. After 24 hours, the cell began to contract, and by 48 hours, they were contracting in synchronization. These are the basic points written in this heading. Now we will move towards the now we see that how cardiomyocytes are cultured. The next heading comes our simulation analysis. Simulation analysis basically a process that uses a mathematical model to analyze the potential outcome of a system or a process. In simulation analysis, the basic points or main points written are this part basically this part of the study basically involves using a simulation software called console software console multiphysics basically is a software that uses numerical methods to model and stimulate physics based problem. As we know, there is a displacement and it is physics, and this software is deal with the physics problems. Now so the emulation involves console multiphysics software to test and understand the performance of the system. The simulation is the cantilever, basically a flexible beam, was analyzed under certain conditions. First of all, simulation setup. These are the subheadings of we are going to explain simulation analysis. The first subheading come simulation setup. First of all, a fixed constraint was applied to the end of the cantilever that does not move, means fixed. This simulates how the cantilever is attached to a surface or the structure in real life. To simulate the effect of cardiomyocytes contracting, a vertical force was applied to the free end of the cantilever. This force was used to make comparison with the experimental results. Even though the cardiomyocytes exert homogeneous stress across the surface of the cantilever, the simulation applied a simple vertical force for ease of comparison and testing. This approach helps analyze how different materials perform under stress. The other subheading is material properties and cantilever performance. I will first tell you the main points of simulation analysis written in this explanation. Then I will show that the screen. The second subheading, the material properties and cantilever performance. The performance of the cantilever is influenced by the material it is made from. The key material property that affects its behavior is a Young modulus, which is a measure of the material's stiffness. A high Young modulus means that material is stiffer and resists deformation better when a force is applied. Basically, in simulation analysis, different materials combinations for the cantilever and sensor were tested. The goal was to demonstrate the benefits of using an SU8 cantilever combined with the monocrystalline silicon sensor. From the next subheading is comparative analysis of materials. In this analysis, there are three different materials combinations were analyzed in the simulation, and the first one is model. First one is model A, and second model B, third is model C. In first model A, the cantilever is made up of SU8 and the sensor can be made of gold. In model B, both the cantilever and the sensor are made of silicone. In model C, the cantilever is made up of SU8 and the sensor is made up of silicone. Each model has different dimensions for the cantilever and sensor. If we compare the force sensitivity of these models, then the force sensitivity of the models was compared by looking at how the resistance of sensors changes when the same force is applied to each model. The formula used to calculate the force sensitivity is uh, 1 minute. This is the formula for calculating the force sensitivity SF, where F is represent the force exerted at the cantilever free end and SF represents the change in sensor resistance or force sensitivity. The finding demonstrate that SU cantilever force sensitivity when combined with the silicone strain sensor was 17 times more higher as compared to gold strain sensors. So by comparing the resistance change for each model, as we mentioned previously, model A, model B and model Their resistance change is compared. The simulation showed that SU8 cantilever with the monocrystalline silicon sensor model C means is 17 times more sensitive than the gold sensor in model A. This means that the combination of SU8 and silicon is much more effective at detecting small changes in force compared to gold, making it superior choice for precise measurements in applications like cardiomyocyte contraction testing. uh, this is this is the points written in this simulation analysis. I, I tell you the main points of the simulation analysis, but in this article there so much detail is written, and I tell you the main points. Then now we will move towards the next heading, which is characterization. Now the heading comes as characterization, because when we prepare something, we must analyze it, and its analysis basically its characterization. So in characterization, the points written are the main properties of the sensors, basically by tested by measuring the change in resistance, as we know, and change in voltage across the Wheatstone bridge circuit with the displacement applied to the cantilever as the main variable. In this, a PZT driver was used. We will see the what is the PZT driver below. First of all, move to this. A PZT driver was used to move the cantilever and create a displacement at its free end. The positive driver basically controlled by the function generator and the power amplifier, and the drive voltage was increased, and drive voltage was increased gradually from 20 volt to 160 volt. The performance of the silicon strain sensor was evaluated based on the output signals, electrical signals, and the displacement. Basically, there is a linear relationship between the displacement applied to the cantilever and the change in sensor resistance. At 10 micrometer and 100 micrometer displacement, the sensitivity change rates were 1.33 multiplied 10 to the raised power minus 4 and 12.38 multiplied 10 to the raised power minus 4 respectively. When the applied load was removed, the silicon sensor returned to its initial state without significant hysteresis, means delay in response. By comparing the experimental result with the simulation data, the gauge factor, basically you will see the details of PCT driver and gauge factor uh, below. First of all, gauge factors and force sensitivity of a sensor were found to be 89.004 and 0.43 micro per Newton respectively. These values were very close to the target values of 93.6 and 0.46 micro per Newton, confirming the sensor's accuracy. Um, the change in sensor output voltage, basically there is a change in sensor output voltage at small displacement like 0.6 micrometer, measured using a laser vibrometer. Now these all setups can be seen with the help of diagram, which is not here. Even at small displacement of 0.6 micrometer, the change in output voltage was significant at 45.34 plus minus 11.72 MV. This change corresponds to detecting forces as small as 0.02 micronewton, which highlights excellent sensitivity of the system. The system can detect very small forces even in noisy environments, providing reliable measurements. As we read the term gauge factor basically is a measure of sensitivity of a strain sensor. It indicates how much the resistance of the sensor changes when it's subjected to strain, force, or displacement. For the formula for the gauge factor can be this: gauge factor is equal to equation number 1, delta R over R, where R stands for the starting resistance, and delta R for resistance change, and E for strain sensor strain. Basically, a higher gauge factor means the sensor is more sensitive to change in force or strain. Now, we also read the term PCT driver. Basically, it is piezoelectric transducer driver, and it is a device used to control a piezoelectric transducer. PCT materials basically change shape or deform when an electric voltage is applied to them, and then they generate an electric current. When they are physically deformed, this behavior makes them useful for applications that require precision movement or vibration control. That's why PCT drivers use this sensor application. A PCT driver is responsible for applying a voltage to the PCT material and causing it to generate a desired movement or displacement. Now we will move the next heading of this review article, which can be drug testing, and this is all about the characterization. Now the last one heading is just testing. As we know that after the device we have to test it with different drugs. First of all, to test the device ability for drug screening, we use two well-known drugs, verapamil and isoproterenol, which affect the way heart cells cardiomyocytes means or cardiomyocyte contract. These drugs basically influence the excitation contraction coupling process, which controls heart muscle contractions. And verapamil is basically a calcium channel blocker that reduces the amplitude and rate of cell contraction. As the concentration of the verapamil rises, both the concentration force and beat rate of the heart cell decrease significantly. Isoproterenol, and the second drug, which is isoproterenol, is a sympathetic. Isoproterenol is basically a drug that increases calcium ion movement in heart cells, leading to a positive inotropic effect, increased heart muscle strength. Basically, when cardiomyocytes are treated with increasing concentrations of isoproterenol, the contractile forces and heart rate gradually increases. At a concentration of 1000 nm, the heart cells start growing, showing signs of tachycardia, which is a state of fast heart rate, which agree. Now we see the effect of both drugs, verapamil and isoproterenol. Um, and I tell you the main important points written in this. Like, now we will move towards the conclusion or result. This is all the detail of the drug testing, and I tell you the main points only. Now we will move to results and discussion. In short, conclusion is, by replacing the traditional metal strain sensor with a single crystal silicon strain sensor, we greatly improved the sensitivity of detecting the force of cardiomyocyte contractions. The experiments shows that single crystal silicon strain sensor can detect extremely small forces as low as 0.02 micronewton and is nearly 17 times more sensitive than metal sensors. We also use the integrated SU8 cantilever to measure how verapamil and isoproterenol, these two drugs, affect the contraction properties of cardiomyocytes. That's it. Now I will show you the whole article that I explain with only main points and basic. And I'm not I have not read all this article. Now I will show to the whole article. First of all, there is abstract. This is all the details of abstract. Then introduction. Then finally comes the application, main focus of this review. These are the details of this review article, which I explain with some basic points. This is the structure and fabrication processes. I hope you will understand. Then characterization comes, simulation analysis, cell culture, drug testing, and then at last result and discussion. That's it. This is all. Thank you.